Good morning students. Today we are going to study about in 4th semester Animal Physiology, Cellular Metabolism and Embryology, Unit-1. In Unit-1 the first topic is process of digestion and assimilation. In digestion, we all know that the digestion is the conversion of the complex solid non-diffusible food substance into the simple. It includes both mechanical and chemical. Mechanical means cutting, chewing and churning, and next chemical is by the enzymatic reactions which occur in our body. Next, the complete digestion is occurring some steps like digestion in buccal cavity, stomach, small intestine and large intestine. In buccal cavity, we all know that the buccal cavity performs two major functions, that is mastication of food and the facilitation of swallowing, which means also called as deglutition. And next, teeth and tongue will help with the help of the saliva masticate and mix up with the food thoroughly. And next, mucus in saliva helps in lubricating. It gives the lubrication for the food to churning the mastication and adhering the masticated food particles into a bolus. And next, the saliva secreted into the oral cavity contains the electrolytes such as sodium, potassium and some enzymes such as the salivary amylase, ptyalin and lysozyme. Next, the chemical process of digestion is firstly it is initiated in the oral cavity, also called as the buccal cavity, and the first steps we are going to see is the hydrolytic action of carbohydrate starch, which means it is a splitting enzyme. It splits. The enzyme is called as the salivary amylase. About 30% of starch hydrolyzed here into a disaccharide called maltose by the enzyme ptyalin. Lysozyme present in the saliva acts as an antibacterial agent which prevents the infection. Here in this reaction we can also start with the reaction of salivary amylase, which is pH 6.8. It converts into maltose plus isomaltose plus alpha dextrins. Next, the digestion in stomach. Almost 4 to 5 hours the stomach stores our food. The food is mixed thoroughly with all the gastric juice of the stomach by the churning movements, which means as muscular wall movements, and the product final product is called as the chyme. Next, HCl provides the acidic pH of 1 to 8, which is optimal for the action of pepsin. It also kills the microorganism ingested along with the food. The proenzymes of gastric juice are called pepsinogen and prorenin. On exposure to HCl, they are converted into the active enzymes like pepsin and renin. Like respectively, pepsin convert the pepsinogen into pepsin. Here we can see the reactions with the help of the HCl. Pepsinogen with the help of HCl converts into pepsin. Protein into peptones, prorenin into renin, casein, milk protein which is present. These all are steps present in only in infants. Paragisin, milk protein. Next, the small intestine. The various type of movements are generated by the muscularis external layer of the small intestine, and these movements help through mixing up of the food with bile, pancreatic juice and intestinal juice in the intestine, and hereby their digestion will facilitate. The mucus along with the bicarbonates from pancreas protects the intestinal mucosa from the acidic medium. In previous step we have studied that HCl with the pH of acidic pH of 1 to 8. Here we are studying that the bicarbonates from the pancreas protect the intestinal mucosa from the acidic medium and provides an alkaline medium with a pH of 7.8 for enzymatic activities. The adrenal cells of the proximal part also produce large amount of bicarbonate to completely neutralize any gastric acid that passes further down into the digestive tract. In small intestine there are 4 types of digestions we can see, that is digestion of proteins, fats, carbohydrates and nucleic acids. In small intestine, these are the reactions we can observe in the small intestine with the help of pancreatic enzyme. Starch plus H2O gives rise to maltose. Maltose plus H2O with the help of maltase enzyme gives rise to glucose. Proteins plus water gives rise with the help of trypsin gives rise to pepsid. Peptides plus H2O with the help of peptidases gives rise to amino acid. This is the small intestine enzyme. These are the pancreatic enzyme we can observe in the digestive reactions in the small intestine. Next, the proteins. We all know that the trypsinogen, chymotrypsinogen and pro-carboxypeptidases are the inactive enzymes. When these are activated by the enzyme enterokinase secreted by the intestinal mucosa into the active trypsin, which in turn activates the other enzymes. Firstly, with the help of a enterokinase, the trypsinogen is activated into the trypsin, and next these trypsin activate the next one, trypsinogen into trypsin, and the end product of the digestion of proteins are amino acids which are formed in the small intestine. These are the reactions we can observe. These are the reaction of proteins with the help of the enzymes in the small intestine. Next, digestion of fats. Bile cells of the bile help in the emulsification of fats, which means the fats will be break down into the small particles. Bile also activates lipases of pancreatic juice and intestinal lipases. These lipases are act on emulsified fats and convert them into the fatty acids and glycerol. Here we can observe fat with the bile and agitation. It is converted into the emulsified fat. Next, this emulsified fat is with the help of the pancreatic lipase. It is converted into the fatty acids and 2 monoglycerides. Next, digestion of carbohydrates. Carbohydrates in the chyme are hydrolyzed by the pancreatic amylase into disaccharides. Intestinal disaccharides act on the disaccharides and convert them into the monosaccharides. We all know that here the complex molecules is breaking down into the simpler molecules. That's why the intestinal disaccharides act on the disaccharides and convert them into the monosaccharides. Here, polysaccharides, the starch and glycogen which is present in our food, it is converted into the maltose with the help of the amylase enzyme, and next this maltose is a disaccharide, and next it is converted into the glucose which is a monosaccharide with the help of the maltase. Likewise, sucrose and lactose, with the help of the sucrase it is converted into the breakdown into the glucose plus fructose, and by the lactose with the help of the enzyme it is converted into the glucose plus galactose. Next, the digestion of nucleic acids. The nucleic acid which is present in our food of the pancreatic juice, it acts on the nucleic acids and it converts into nucleotides and nucleosides. These both are the nucleosides of the intestinal juice, convert them into the pentose sugars and nitrogenous bases. Here we can observe nucleic acid. With the help of the nucleases, it converts into nucleotides. Nucleotides converts into nucleosides. Nucleosides converts into sugars plus bases. Next, digestion in large intestine. The waste products, in the entire digestion, the waste products include the undigested parts of the food, fluid, and the older cells of the lining of the gastrointestinal tract. The large intestine absorbs the water and changes the waste from liquid into the stool. Next, peristalsis helps move the stool into the rectum. In rectum, the lower end, which is the lower end part of the large intestine, the rectum function is to store stool until it pushes out the stool out of your anus during a bowel movement. Next, absorption. What is absorption? The process of absorbing the end products of digestion into the bloodstream or lymph from the intestinal mucosa. During absorption, the nutrients are carried to the bloodstream. In assimilation, it is the process of carrying. In absorption, we can observe the process of absorbing the end products, but in assimilation, we can observe the process of carrying the absorbed simple molecules to the different organs of the body with the help of the blood vessels to build the macromolecules. These macromolecules are responsible for cell growth and the development, and in this assimilation, nutrients are consumed from the bloodstream. The next topic, respiration, pulmonary ventilation and transport of carbon dioxide and oxygen. In the pulmonary ventilation, we all know that it includes the two processes called the inhalation and the exhalation. In the inhalation, the muscle will contract and expands the thoracic cavity, and it also decreases the intrapulmonary pressure, draws air into the lungs. And next, in exhalation, the muscles will relax and decrease the thoracic cavity volume, and it also increases the intrapulmonary pressure, forces air out of the lungs. Next, here we can also observe the gaseous exchange also occur. Oxygen enters into the bloodstream through the alveoli, which is present in the lungs. Carbon dioxide exits the bloodstream into the alveoli. Next, the regulation, neural control. Respiratory centers in the brainstem regulate breathing. Next, chemical regulation. Chemoreceptors monitor blood gas levels. Permanent ventilation facilitates the exchange of gases necessary for cellular respiration and is regulated by neural and chemical mechanisms. Here we can observe this is the picture of inspiration and this is the picture of expiration. In inspiration, we can observe when we take the air into the lungs, the thoracic cavity expands, the internal costal muscles contract, and this is the diaphragm, this will contract. And next, during expiration, when we leave the air, thoracic cavity reduces and the external intercostal muscles, these muscles, the muscle between these two bones, is called as intercostal muscles, will relax and diaphragm relaxes. Next, the transport of oxygen is done in two steps. The binding to hemoglobin. Oxygen binds reversibly to the iron atoms which is present in the blood, called as hemoglobin molecules, which forms oxyhemoglobin. Each hemoglobin molecule can carry up to the 4 oxygen molecules. And dissolved in plasma, a small amount of oxygen remains dissolved in the plasma, contributing minimally to the oxygen transport. Next, transport of carbon dioxide. Dissolved in plasma, carbon dioxide dissolves directly in the plasma, forming the carbonic acid. This dissolved form of carbon dioxide is transported to the lungs. And next, bicarbonate formation. Carbon dioxide diffuses into the red blood cells where it combines with the water to form carbonic acid. Next, binding to hemoglobin. A small fraction of carbon dioxide binds directly to the amino groups of hemoglobin, forming carbaminohemoglobin. Next, chloride shift. To maintain the electrical neutrality, the chloride ions move from the plasma into the red blood cells as the bicarbonate ions are formed. Next, transport of hydrogen ions generated during the formation of carbonic acid bind to hemoglobin or other buffers to help maintain blood pressure. This is the picture. Oxygen, air, alveolar wall. The red blood cells carry the oxygen molecules, and here carbon dioxide will be out into the air. Next, structure of human heart. The human heart is about the size of a human fist and is divided into 4 chambers. There are 2 ventricles and 2 atrium, and the ventricles are the chambers which help to pump the blood, and the atrium are the chambers that receive the blood. Among these, both the right atrium and the ventricle make up the right heart, and the left atrium and the ventricle make up the left heart. The structure of the heart also houses the biggest artery in the body, called as the aorta. The right and left region of the heart is separated by a wall of muscle called the septum. The right ventricle pumps the blood to the lungs for reoxygenation through the pulmonary arteries, and the right semilunar valves close and prevent the blood from flowing back into the heart. Then the oxygenated blood is received by the left atrium from the lungs via the pulmonary veins. Next, this is the structure of the heart. This is right atrium, left ventricle, right ventricle. This is the aorta. This is the superior vena cava. This is the pulmonary valve. This is one is called as the tricuspid valve. This is inferior vena cava. This is pulmonary artery, pulmonary vein, and this is mitral valve, and this is called as aortic valve. Next, let us know the function of the heart. The function of the heart in any organism is to maintain the constant flow of blood throughout the body. This replenishes oxygen and circulates nutrients from the cells and tissue. These are the main functions. One is that to pump the blood throughout the body, and it also delivers oxygen, hormones, glucose and other components to the blood, including the human heart. And it also ensures the adequate blood pressure which is to be maintained in the body. And there are also two types of circulation, that is pulmonary circulation, and the second one is the systemic circulation. In pulmonary circulation, it is a portion which means it carries deoxygenated blood away from the heart to the lungs and then bringing oxygenated blood back to the heart. This is called as pulmonary circulation. And in systemic circulation, here oxygen blood is pumped from the heart to every organ and the tissue in the body, and the deoxygenated blood comes back again into the heart. Now the heart itself is a muscle, therefore it needs a constant supply of oxygenated blood. This is where another type of circulation comes into play, that is called as coronary circulation. Coronary circulation is the essential portion of circulation where the oxygenated blood is supplied to the heart. This is the important as the heart is responsible for supplying the blood throughout the body. This is the picture. We can see here lungs. The oxygenated blood from the lungs enters into the heart, and here the oxygenated blood from the lungs come here through this, and here the deoxygenated blood. Likewise, the oxygenated blood from the lungs goes into the heart, and from the these deoxygenated blood goes into the lungs likewise. Excretion. Structure of kidney. It has a convex and concave border. Towards the inner concave, a notch called as hilum is present through which the renal artery enters the kidney and the renal vein and ureter leave. The outer layer of the kidney is a tough capsule, and on the inside the kidney is divided into outer renal cortex and the inner renal medulla. The hilum extends inside the kidney into a funnel-like space called the renal pelvis. The renal pelvis has projections called calyx. The medulla is divided into medullary pyramids which project into the calyces. Between the medullary pyramids, the cortex extends as renal columns called as columns of Bethany. The kidney is made up of millions of smaller units called nephrons, which also a functional unit. This is the structure of the kidney. This is the renal column, this is the medullary pyramids, this is the calyx, and this is the renal artery, this is the renal vein, this is the pelvis region, this is the ureter, and the outside layer is called as the renal capillaries. The inner layer is called as the cortex. The main functions of the kidney is to filter the main function of the kidneys is to the filtration and the reabsorption like essential substances like water, glucose, ions, and the secretions like additional waste products and the ions are actually secreted into the blood, and the excretion, the final urine containing the metabolic waste and excess ions produced and secreted from the body. The countercurrent mechanism. The mechanism that the kidney is used to concentrate urine is called as the countercurrent mechanism. In this we can observe three steps: thick ascending loop of Henle transport to the equilibrium of descending thin loop of Henle. Next, fluid flow. The nephrons involved in the formation of concentrated urine extend all the way from the cortex of the kidney to the medulla and are accompanied by vasa recta. The filtrate flows into the two limbs of Henle's loop in the opposite direction, and therefore the flow of blood cells in vasa recta is also in the opposite direction. Here is the picture of how the countercurrent mechanism is done. The more salt in the continually added by the proximal convoluted tube. Here it is 300, next 400. Here water will be absorbed. This is the entire cycle of countercurrent mechanism.